Hi, I'm Jan Witkowski of Cold Spring Harbor Laboratory. This is the 83rd Annual Symposium, the 83rd Cold Spring Harbor Symposium on Quantitative Biology, and the topic this year is brains and behavior. And I'm delighted to have with me this afternoon Huda Zoghbi from Baylor College of Medicine, who will be talking this evening.
Correct.
So we're going to get a sneak preview.
That's right.
So Huda, your title is enticing, fine-tuning protein.
Fine-tuning protein levels for neurological health. Mm-hmm.
All right.
So what does it really mean? What does it translate to in scientific terms and in practical terms? Mm-hmm. Because this was a broad meeting encompassing all sorts of brain function and dysfunction, I decided to talk about a principle that, in my experience, having been now in research for 30 years, in my experience spans all aspects of neurological disorders that I have discovered, from childhood diseases all the way to adult aging-related disorders. And that's the principle, the overarching principles, that protein levels matter for brain health. So I will cover the topic today by describing what we learned from adult neurodegenerative disease, and then what we learned from studying childhood neurodevelopmental disorders, and how that principle emerged. Actually, for the first 10 years or even 20 years of my career, that was not anywhere on my radar screen, but it was really through the data and the experiments we've done.
And when you're talking about protein levels, are you meaning levels of particular proteins?
Correct.
Or gross?
Correct. So what I will be talking about are examples of proteins that the brain is exquisitely sensitive to their level. And for some of these, a small change in the 10 to 20% range will make the older brain highly vulnerable.
And this is a change of level, not a mutation in that protein.
Well, sometimes it's pure. So some examples I will give are pure change in level without any mutation. And this is some of the recent data we have. And some of it, a mutation will lead to enhanced level and or function of the protein. So you can get there in more than one way. You can get there by a mutation in the gene encoding the protein itself, or you can get there by changing the level of a regulator of that protein, sure. Right? So I'll use some of these examples.
So an example that comes to my mind, which I think on further reflection is not correct, but in spinal muscular atrophy, where you have multiple copies of SMN2,
correct,
you get more protein.
So in spinal muscular atrophy, the SMN2 gene, you actually have multiple copies of it, but there's one copy, the one that's properly spliced, that make the most of the protein. And the mutations in the gene encoding that, when they happen, you inactivate the protein, so you're left with very little protein, and the patients have disease. But their disease is modified by the level of the paralog, the SMN2, so this is SMN1, and if you have very little, then it's a very severe phenotype. If you make a little bit more, you make it, still severe, but more moderate, and then if you have a lot more, you make it milder. So the new strategy for therapy, and that's due to a splicing mutation, a mutation that leads to abnormal splicing. You don't make a full-length protein. So the new therapy, the antisense, allows that exon to be skipped, allowing for more of the protein to make, which rescues the symptoms.
But is that sort of dosage effect of multiple copies the sort of...
No. So let me maybe tell you a little bit about that. So that may be making it clearer. So from the end of, from the neurodegeneration, I studied a very, very rare disease. Nobody probably heard of it. It's a balance disorder. It affects coordination and walking, and it happens later in life, typically when the person is a mature adult. The reason we studied it because it's inherited. It's Mendelian, so it was easy to track. And my thought was, if we learn something from it, it might inform us for the broader class of neurodegenerative diseases. So we did that, and in 1993, Harry Orr and I identified the gene and found it's a repeat expansion, trinucleotide repeat, CAGs, that encode glutamine. And that glutamine, in you and I, we may have 30 glutamines, but in the patients, they typically have 39 or more glutamines. So imagine the difference between 30 and 39. If you just have a little bit more, you're going to now lose critical cell types in the brain that coordinate balance. So we created animal models, we studied that, we identified its protein interactions, and I will summarize 20 years of work in the following discovery. We discovered that really what happens with the glutamine expansion, it stabilizes the protein, makes it a little bit more stable, a little bit more abundant in the cell, a little bit more enhanced interaction with its known normal native partners. So you're basically having a little too much of something. So, and we tested all that genetically, biochemically. We're able to show, we have a readout for its interactions. We can really show that those interactions are enhanced, and if you remove the interactor by 50%, you can now suppress the disease symptoms in animal models. So it told us then, if that's the case, we really need to study what regulates this protein. So we identified an RNA binding protein that regulates it, and this is where we discover you don't have to mutate ataxin-1 to cause toxicity, because when we took out one copy of the regulator, so the RNA binding protein binds the untranslated portion of the RNA and suppresses the level of the RNA, keeping the ataxin-1 protein in check. The ataxin-1 is the protein mutated in ataxia. What we found, if we reduce the RNA binding protein by 50%, then ataxin-1 levels go up by 20 or 30%. And that increase of 20% or 25% of a wild-type protein induce Purkinje cell degeneration and balance problem. How are we sure it's that ataxin-1 25% increase? Because we rely on genetics. We can now take these animals that lack one copy of the regulator and therefore have a slightly higher ataxin-1 and breed them to mice that lack one copy of ataxin-1 to normalize ataxin-1.
Very nice.
And you block the degeneration, and all the symptoms for balance problems disappear. So this really pretty much for us nailed the importance of protein levels. Even a healthy protein can be toxic if increased by 20 or 30%.
So I can see that, I'm sure you'll come onto this, that this offers therapeutic opportunities.
Correct.
But it also surely expands the number of ways in which one can get a neurodegenerative disease.
Absolutely. Which is the second middle part of the talk. You got it. So when we saw that, we immediately recognized this offers a therapeutic opportunity. How? Because if we can find regulators of ataxin-1 that can be inhibited safely, and for which you can design a small molecule, then we could perhaps develop a therapeutics. And we set out to search for those using genetic screens in human cells and in collaboration with Juan Botas in fruit flies. And we identified many candidate regulators that we are pursuing one by one, some of which we're now actually working with a pharmaceutical company to develop a therapeutic for. So that's when it comes to SCA1. But if you observe what else happens around you in the literature for other neurodegenerative diseases, could what we have learned from this ataxia project apply to the more common neurodegenerative diseases? Well, the genetic data tells us yes, because we know that doubling the levels of normal synuclein can cause Parkinson's disease. Tripling the level of normal synuclein can give you earlier disease onset. Doubling the amyloid precursor protein gene gives you dementia. People with Down syndrome who have a duplication due to trisomy 21 have onset of Alzheimer's at 35.
Yes.
So we know for these that genes and proteins levels matter. You don't have to mutate any of them.
That's just what I was going to just confirm with you, that for example, in the Downs case,
Right, it's normal. It's just
it's just
extra
normal.
APP. And we know it's the APP gene because there are a couple of individuals who have the trisomy, but they lack the APP gene and they don't get the Alzheimer. So Down individuals may get dementia at 35 or 40. So it's really important for us to understand and figure out how to regulate APP levels.
I mean, going off on a little sidetrack, why hasn't this sort of been thought of before? Have people been so fixed on the idea that in a neurodegenerative disease or any certainly inherited disease, you have a mutation in a gene that produces a mutated protein, and have rather ignored the fact that these fairly small differences in protein levels can have profound effects?
I think people have thought about it but have not pursued it necessarily as aggressively as needed. Clearly the duplications and the triplication of cellular, those were studied genetically and those were Mendelian inherited, although it's wild type protein. I think people are coming around now to realize another protein, tau, where regulatory mutations or mutations that change the isoform variants can also drive degeneration. So this is where the lesson of rare ataxia got me closer to understand now perhaps at least one approach to think about these common degenerative diseases. Let's find the regulators of these proteins and let's think of ways maybe we can slightly lower the levels of these disease-driving proteins, and that might protect people. And the value of that, because it comes back to your question, there are many ways we can get neurodegeneration. You're absolutely right. If you're a person with a duplication of APP, you're going to get early Alzheimer. But you may be a person with healthy APP, a person that doesn't have any duplication, but may have a variant in a regulator of APP. That's going to slightly increase APP. That might put someone at risk at 70 instead of 80. So finding the regulator is going to teach us more about the biology of these proteins, but also it's going to help us interpret DNA sequencing data. Right now we're hit with so many variants. We have no idea which ones to study and why, and we can put them in animal models and we might or might not get a phenotype, but at least if you had the framework that these 70 genes are known to regulate synuclein, these 100 regulate APP, and so on and so forth, then at least if you got variants in them, you have a functional assay.
I was talking to Joshua Gordon this morning, and he was talking about the way he hopes NIMH will go forward, and we were talking about GWAS studies and the large number of genes you might find in an implicated region. So have people yet begun looking in sort of GWAS studies for regulatory genes of the sort that you're talking?
We just started because we just really began to identify a large number of regulators that we're validating, and we are crossing these now with GWAS studies for Alzheimer and Parkinson, and we're beginning to find overlap, but we still have to do more. This is just the beginning. So we're just beginning to do that. For some, we're collaborating with Allison Goat. For other, we're using existing data that are publicly available.
Can you say you're searching sequences for, do the sorts of regulators you're looking for they must have sort of key signatures that enable you to say, well, this sequence is for a regulator X or...
So I'll give you an example. We might have a kinase for a protein, and if you phosphorylate that protein, you make that protein much more stable. For example, you prevent its degradation. So that kinase becomes a candidate where a gain of function mutation may put someone at risk for neurodegeneration, whereas a loss of function heterozygosity might be neuroprotective. So this is the example.
This sounds a little bit depressing when I think of all the kinases there are and all the phosphatases and think that any one of these might be
a candidate. Absolutely. So I think that for the majority of the people, majority of us or people don't have necessarily the risk to have a duplication of synuclein or APP because we don't have the family history. But it's going to be something else, right? Because the pathology incriminates these proteins. These are at least some of the more common. If you took 100 people with Alzheimer's, the vast majority of them will have abnormal APP accumulation or A beta accumulation, abnormal tau phosphorylation. So you know they're effectors at some point in time, but there's no mutation in them. That's why I think the regulators are important. Right,
so you can recognize effectors that are normal.
Right.
But producing a phenotype, clinical phenotype. And then you say, well, it's not that, it's the level of the protein, not the mutation or the
correct. It's what it's affecting. That is correct. So I didn't finish this. I should have followed up for you. In the case of the ataxia, you remember the regulator I mentioned to you, that if we took one allele, we saw elevation of ataxin 1 levels, and we show in the mice degeneration. We then found people with haploinsufficiency, with deletions or inactivating mutations of one allele, and these people have childhood ataxias, and they have developmental disability and other neurological problems. But if they have even a milder mutation, not total null allele in one allele, if they're haploinsufficient due to just missense mild mutation, they have a late onset balance disorder. So you see the gradation of the phenotype.
And certainly the therapeutic opportunities provided.
Actually, it's more optimistic this way, because
if
you actually knew that this is a big driver of disease, finding then such regulators, studying them, and finding way to elevate them or decrease them, right, is going to be far more valuable.
Yeah, I mean, that's almost sort of standard pharmacology towards levels of things as opposed to replacing a mutant gene, for example.
Exactly. So this is all with neurodegeneration. Now, if we have a couple more minutes, I'll tell you about childhood diseases. So as you know, I've always been interested in Rett syndrome, when we discovered it's caused by mutation in the gene methylcytosine binding protein 2, MACP2. Well, that protein turns out to be extremely, its function is extremely critical for the brain, and levels really, really matter. So if you have a mutation that totally inactivates the protein in a male, because it's on the X chromosome, sadly these males will be severely affected and they will die early. But if you have a mutation that's milder, that male will survive and will have neurological function. And depending on the severity of the mutation, their phenotype could be early autism, hyperactivity, or a little later, if it's milder mutation, could be schizophrenia, juvenile onset schizophrenia, or other behavioral problems. So here again, you see gradation from death in the first year of life, with a null allele, all the way to milder psychiatric symptoms if the mutation is milder. And on the other side of the coin, of course, if you have it in a girl, they're mosaic, it's on the X chromosome, so 50% of the cells lacking a functional allele will give Rett syndrome, which, as we know, affects all aspects of brain function. On the other side of the coin, if you double the level of the protein, you get a progressive neurological syndrome. And if you triple it, you get even a more severe phenotype. So here again, we're seeing that level function phenotype relationship.
So I think we should finish in a moment. So the brain presumably is special in this case. I mean, nerve cells are particularly sensitive.
So far, that's what we're seeing. We're not noticing the effect of these protein in dividing cells. We're seeing that either in neurodegeneration or in the childhood diseases like Rett syndrome and the MACP2 duplication. We're seeing those effects manifest in brain. So it seems that brain cells that once mature and they don't divide, they're probably much more vulnerable to change in protein homeostasis. You need to get everything right.
I mean, I seem to remember that the trinucleotide repeat disorders are preferentially brain related.
Right.
And is this the sort
At
of
least the coding regions, the ones that affect the protein specifically, the ones that are
that's
within
true.
the coding region, you're right. They're brain specific. There may be some peripheral phenotype in the spinal bulbar muscular atrophy, but that's really not what drives the disease. It's the brain specific phenotype that drives, or the nerve cell, because here it's the spinal cord. And however, there's some that affect muscle and so on, but these are outside the coding region. Yeah.
Well, fascinating.
Yes.
Fascinating, and I said it was a bit depressing thinking of all the extra things that could go wrong.
Right.
But I think the optimistic side is much better.
Right. No, I think that I find it more optimistic if we know now that protein levels matter. That means we have an opportunity to find things that regulate these disease-driving proteins, and any such opportunity could be translated into therapy, whether small molecule, antisense oligonucleotides. I think new libraries can be used to almost target any protein. You don't anymore have to have an enzyme to have a therapeutic. I think the new libraries that one can use, screening billions of compounds, one could really almost regulate or manipulate any product, if you will.
Well, that seems a really good moment to stop. Thank you very much, Huda.
Okay, thank you.